Bueno, buenas tardes. Gracias por su presencia al seminario de la serie de Frontiers en Genomics. Y realmente es un placer para mí presentarles al doctor Sean Cutler. Él estudió su licenciatura en la Universidad de Toronto en antropología y después realizó su maestría también en la Universidad de Toronto en botánica y su doctorado en Stanford, donde ya en términos generales es ciencias biológicas, pero enfocado ya más hacia la investigación en plantas. Y después realizó su un postdoctorado en el Scripps Research Institute en La Joya, para después convertirse en profesor asociado en la Universidad de Toronto y regresar a Riverside como profesor asociado en... Perdón. En el 2006 como profesor asociado en Riverside. Se promovió en el 2010 a profesor, y en el 2019 ya es un profesor distinguido de la Universidad de Riverside. Ha recibido diferentes premios, de los cuales me gustaría resaltar que del 2017 al 2022 ha sido uno de los investigadores más citados en el área de plantas. Es miembro de la Academia Nacional de Ciencias de Estados Unidos desde el 2018. Recibió un premio como investigador distinguido de la Asociación Internacional de Crecimiento de Plantas y también recibió temprano en su carrera un reconocimiento como un científico joven por Valiant Biosciences. Y Sean ha contribuido notablemente al entendimiento de las bases moleculares que controlan la resistencia a sequía en plantas. De hecho, uno de sus trabajos más relevantes fue precisamente tratando de elucidar cómo ABA puede modular el cerrado y la apertura de los estomas para modular precisamente la pérdida de agua en las plantas, y de ahí le llevó a descubrir precisamente uno de los receptores para ABA que modulan este fenómeno. También ha contribuido notablemente al uso de nuevas moléculas con aplicaciones agroquímicas, precisamente tratando de encontrar aquellas que puedan modular estas funciones para tratar de controlar un poco los efectos deleterios de la sequía en las plantas. Y en todas estas áreas pues ha publicado en artículos muy relevantes. Y también a partir del conocimiento del receptor PIR1, que fue uno de los primeros que encontró, bueno, está tratando de diseñar receptores a partir de ese para poder ser modulados con medios químicos, otra vez con el mismo objetivo de regular la resistencia de plantas a la sequía. Y otra de sus contribuciones es precisamente seguir probando estas librerías, estos péptidos y moléculas bioactivas con el mismo fin. Y en todas estas áreas realmente Sean ha hecho contribuciones muy importantes, y bueno, hoy nos va a hablar de los avances recientes que tienen en la función de ABA, nuevas funciones de ABA en plantas. Y Sean, thank you very much for accepting and being here with us today.
Thank you. Okay, well, thank you for having me here and for that introduction. I am going to tell you all about work that my lab has been doing over the last 7 or 8 years that's been focused on taking some of what we learned from studying how the plant hormone ABA, abrasic acid, works and does what it does, and building on that to try and engineer new traits that could be useful for plant biotechnology. And that involves really trying to re-engineer signaling pathways so that we can do new new kinds of things with them. And I'll explain how we got to where we were, but I wanted to just start off with a little image of kind of where this direction in my lab started, before we go into the details of the background. So in the US, there is a funding agency that is called DARPA. It's for the Defense Advanced Research Projects Agency. And they're very famous for funding all kinds of crazy ideas and often putting a lot of money into crazy ideas. And many of them have turned into really important discoveries. They're famous for funding the first version of the internet in the 1950s, and many other really important inventions have come out of this with almost moonshot type projects that they like to put out there. And so about 6 or 7 years ago, there was a call that came out, and the call was not exactly for what I wanted to do, but they wanted to engineer plants that could perceive 3 different signals, separate signals, like it could be any type of signal, they didn't really care, and then convert those into 3 different outputs. So in this little cartoon here, or it's not a cartoon, but in this little image, it would be that we have a plant, it's going to be planted on some site that has a contaminant that the agency cares about, and if that contaminant is present, the plants would produce some visibly obvious signal that would allow us to detect that the plant is there. This is really just plants as biosensors, but DARPA wanted to be able to have, or their call was such that they wanted multiple inputs and multiple outputs. And so I'm not necessarily myself so interested in planting plants at sites to do biosensing, but to me this more generally illustrated and linked into a problem that I've been interested in for quite some time, and that is really thinking about ways that we can develop tools that could be used for making agriculture more precise. And so this is just a little cartoon I took from the web somewhere. It's not a perfect representation, but it'll serve to make the point that I'd like to make, and that is that there is increasingly more and more data that is available to farmers as they grow their crops. And a lot of the decisions that get made that impact final yield are sort of hardwired. So you have to pick which seeds you're going to plant. You do have control over what sorts of chemical agents you apply, but at least on the genetic side, things are very fixed. And so we've been interested, for example, I'll talk very briefly about this, in trying to develop chemicals that you might be able to use to mitigate the effects of drought if some of this data was telling you there might be a drought in the near future. That's not really the point of my talk today, but the bigger issue here that I'm interested in is thinking about technologies and biological processes that we need to understand that will allow us to intervene in plant growth and development and responses to the environment for the benefit of agriculture. And when my lab started thinking about this and going down this path, we were very interested in the plant hormone abscisic acid, or ABA. This is one of many small molecule growth regulators, or plant hormones, that control all sorts of aspects of plant growth and development. ABA has many, many functions, so I'm oversimplifying tremendously here, but probably one of its more important roles is in regulating the aperture of these small pores that are on the surfaces of leaf, that cover the leaves, and they are the valves that control whether or not water is leaving the leaf and whether or not CO2 can get in. And ABA acts as an integrator in some ways. When water levels are low, this hormone level shoots up. It causes the closure of these guard cells, along with a suite of other transcriptional and physiological changes that help the plant acclimate to the changing water conditions, and other stresses as well. And so when plants are unable to synthesize this hormone properly, they will wilt catastrophically, or wilt much more easily and eventually die than their wild-type counterparts, because they're missing a key part of the signaling machinery that's required to communicate problems about the environment to the physiological responses in the cell, and particularly the opening and closing of these guard cells or stomata. And on the flip side, if you treat plants with exogenous ABA, so if you just spray them with ABA, you force the closure of these of the guard cells. That reduces the amount of water that they're transpiring through their leaves, and it actually buys you time before they start to wilt catastrophically, because they're basically put in a kind of stasis. And since my talk isn't really going to be about this whole idea, I won't say too much. But where my interest in this hormone started was both in trying to understand how it does what it does, how it controls plant growth, but also trying to maybe come up with chemical tools that might be used to intervene during a growing season for some benefit to agriculture. And when I started, we didn't know much about ABA. In the 1980s, a number of Arabidopsis geneticists had done many screens had been done to identify mutants that couldn't respond to this hormone or that wouldn't make it at the proper levels, like the one I showed on the slide before, which was a tomato plant that was wilting. And little bits and pieces came out of that, and some of them looked interesting. There were transcription factors, there were a kinase, there was a phosphatase, but it was really a patchwork of molecular components that didn't fit together. And there was, most notably, there was not a real receptor that the community agreed was actually a receptor. It turns out there isn't a receptor, there's a very large family of receptors. And the reason that geneticists had had such trouble identifying these components of the signaling pathway was because there are so many of them. If you knock any one of those receptors out, we now know in hindsight, the phenotypes are at best very modest. And for the most part, it's extreme. You can't detect any evidence that there's a problem with ABA signaling. So geneticists were blind to the receptors and other components of this signaling pathway. So when I started my lab, we speculated that perhaps the redundancy could be an issue, and we needed to take a really different approach than what geneticists had been doing. And so we took a chemical biological approach, and we screened through thousands of compounds, and we identified a synthetic molecule that we called pyrrobactin that would mimic the action of ABA. And this is now bordering on ancient history, so I won't go through all the details. But what we discovered was that if we did a genetic screen using this as our chemical molecule instead of the native hormone, that we were able to identify a gene that had never been discovered through those classical forward genetic approaches. And I'm cutting a very long story short, and I'm going to tell you what we learned and what the years of work on this from my lab and many other labs told us. So in gray here is a soluble receptor. ABA is here in the middle. This is from these are an image generated from coordinates from one of the many crystal structures that exist. And this pink blob here is a protein phosphatase. So what we learned is that when ABA levels go up, it binds to the receptor that we identified through our genetic analysis using this chemical probe. That triggers a conformational change in a loop that's in a small pocket that's surrounding in a loop that flanks this small ligand binding pocket. That conformational change allows the receptor to form a very stable complex with the pink blob here, which is a specific type of phosphatase called a type 2C protein phosphatase. And when the ABA activated receptor forms this complex, it's inhibiting the enzymatic activity of this phosphatase, so it's no longer able to dephosphorylate its normal substrates. This is an illustration of what's known biochemically as chemically induced dimerization. And this term, CID, or chemically induced dimerization, is going to be really critical to my talk, so I will come back to it again. I am not really, we're going to leave ABA behind in the next, probably after this slide or two, but I just want to kind of put it all together before we pivot to the more engineering. So the way this system works is that the gene we identified that gave resistance to this molecule we called PIR1, and it's part of a very large receptor family, as I told you, where there's tons of genetic redundancy. Normally, there's a central stress-activated kinase, and a family of them that are called the SNRK2s or the SNRKs, And they are the master regulators that control the activity of downstream factors through phosphorylation. They have to be activated by phosphorylation on one specific residue in what's called an activation loop. And normally, the pink blob that I showed you, the phosphatases, are dephosphorylating the phosphate on that activation loop. So the default state of this pathway is to be off because there's the phosphatases keeping the right master regulators off. When ABA comes along, it binds to this receptor protein and its other relatives. That leads to the formation of this stable complex, inhibits the phosphatase activity, and then that allows the activated forms of the kinases to occur. There are probably some kinases that may also activate these separately, but in a test tube, these kinases will auto-activate on their own. So if you put just these three pieces and a target protein in a test tube, you can reconstitute the hormone-mediated phosphorylation of one of the regulators downstream with these three pieces. And these downstream targets include transcription factors and many other proteins that regulate and execute ABA's outputs. So the bottom line from all of this is that our work and the work from other labs demonstrated that ABA it's sort of at the top of the ABA signaling pathway is this unusual mechanism of chemically induced dimerization where the hormone is stabilizing the formation of a complex that is normally keeping the pathway repressed. So it's like a classic de-repression type pathway that may be familiar to many of you. Okay, so it turns out that if you're going to compare plant signal transduction and animal signal transduction, there are some similarities. Plants do have some membrane localized receptors that initiate kinase cascades, but actually for the most part, at least for the major hormones that plant biologists care about, the sensing systems are rather quite different. They involve soluble proteins and they frequently involved this chemically induced dimerization mechanism. So the first real beautiful example of this came from work from Mark Estelle and many others characterizing how the plant hormone auxin works. And to cut a long story short, this is an SCF ubiquityl ligase complex with an FBOX protein in it called TIR1. And TIR1 and a co-repressor will form a complex together with auxin sitting in the middle is what they call a molecular glue. So auxin's function is to stabilize a complex between this SCF complex and one of the downstream target proteins. Once the complex is stabilized by a chemically induced interaction, that leads to the ubiquitination of this green blob here. It's a transcriptional repressor, it gets degraded, and that initiates downstream signaling. So this is the sort of first canonical example. And all of these other hormones, which are some of the major regulators of plant growth and development, involve a form of chemically induced dimerization. The details are a little different. Most of the times it's actually that the interaction is regulating proteolysis through ubiquitination. But in the case of two hormones, the one I've told you about, ABA, and another important plant growth regulator, there's a conformational change that ends up leading, in the case of ABA, to a change in enzymatic activity. So the bottom line from this is that this mechanism of chemically induced dimerization is a really critical, common feature or theme in plant signal transduction. So again, I'm going to veer away from ABA real quickly, but I just wanted to point out, circling back to this theme of developing tools for intervening with growth and development and stress responses during agriculture, that one offshoot of this work was that we discovered that the receptor family can be chemically controlled, that we could develop or discover or design new molecules that regulate the activity of these receptors. So basically agonists or antagonists. And one part of my lab has invested quite a lot of effort in designing a mixture of discovery and design of molecules that are much better activators of the receptors than that first molecule, pyrrobactin, that I told you about. None of this has led to anything commercially important yet, but a couple of the big agrochemical giants have invested a lot of effort in trying to develop these kinds of molecules. So I'm going to switch now to more of a synthetic biology view and where this project has gone off, and take us towards trying to engineer genetic circuits in plants where we are able to program them to respond to chemicals at will. And a lot of this, as I've already alluded to, is going to link into this chemically induced dimerization mechanism. And the reason that we're so obsessed with CID in my lab and in the talk I'm giving today is because it's a really, really beautiful biochemical mechanism for building genetic switches. So the way we actually discovered the pink blob in this picture, the phosphatase, was we did a yeast-2 hybrid experiment where we used the receptor, what we now know to be the receptor, we didn't know at the time, as bait. We added our chemical ligand, and we were able to fish the phosphatase out as a protein that would bind to the receptor in a ligand-dependent fashion. And that's basically what's being illustrated in this cartoon here. We can create chemically regulated genetic switches, which is what the yeast-2 hybrid uses, but you can transplant this into other systems. But you can also do things with chemically induced dimerization, like reconstitute two-split proteins. I think I have one slide where I'll show you us doing that with luciferase, so you can have, ligand-mediated production of luminescence. There's many, many variations on this, and there's been a lot of development in this area because in the 1980s, the first chemically induced dimerizers were discovered that are rapamycin and related immunosuppressant molecules. And so chemical biologists have been harnessing this mechanism for a long time to build chemically regulated processes. It's great. So chemical biologists noticed this, all of these interesting features about plant hormones, and they've used them to engineer different types of chemically regulated systems that are based on chemically induced dimerization. One of them is the auxin-degron system, which was built from that auxin system I told you about. In this case, the addition of auxin leads to the ubiquitination and degradation of a target protein. The Crabtree lab, one of the labs that discovered how the immunosuppressants work and chemically induced dimerization in the 80s and 90s, saw the ABA work, and they built a transcriptional circuit that works in mammalian cells with the ABA receptor and the phosphatase. And the GA receptor, another one of these, was harvested to build a GA-regulated system. And you can actually use these two together in the same cells. These are all systems that have been developed for mammalian cells. And this is great if you work in mammalian cells, but it's not so great if you work in plant cells. The problem is if we stick these pieces back into plants, they're going to activate and interact with the endogenous signaling systems that they normally coordinate. Getting to back to this idea of trying to engineer signal transduction, where this is all coming together is that we've invested a lot of effort over the last 7 or 8 years in both rewiring the ligand binding pocket of the receptor so that, okay, hang on a second. Yeah, we've spent a lot of time, and I'll go through this data and how we got there, rewiring the ligand binding pocket so that we can get the receptor to recognize other molecules besides ABA. This is to create what are called orthogonal receptor ligand pairs. And we've invested a lot of effort in rewiring or hacking the protein interface that the phosphatase uses to interact with the receptor, because what we really want to do is to create a chemically induced dimerization module that's fully insulated from the plant signaling components, but that we can reprogram its ligand binding. So in our fantasy, we can engineer simple modules that are activated by whatever chemical we want off the shelf. So it's not going to go quite that far, but I think we've actually made a lot of progress towards that general idea that I just outlined. And so what we're really trying to do is to create new chemically induced dimerization modules where we're able to reprogram the ligand binding specificity and create new types of entirely chemically regulated switches. Okay, so part of the reason that when that call that I started out came out, part of the reason we thought we might be able to do some of this stuff was because of an experiment we had done years before for a completely different reason. And that was the following. So what we did was, I told you that my lab was trying to make these chemicals that activate ABA signaling, and maybe they could be useful in agriculture. It turns out that the cost of bringing a new molecule to market is very, very expensive. And so we had this kind of repurposing idea where we thought perhaps we could take an existing agrochemical, reprogram the ABA receptor binding pocket so it recognized that agrochemical, and then we would just have to put that engineered receptor back into a plant, a transgenic plant, and with that engineered receptor, we'd be able to artificially activate ABA signaling using a molecule that's already being used by farmers in the real world. So that was the idea, and it worked. It took us many tries, and I learned some hard lessons about protein engineering through this project, but we eventually figured out how to do it properly. And we were able to reprogram the main receptor we work on, which is called PIR1, to recognize this agrochemical that's called mandipropamide, or Mandy for short. It's used currently in crops to control Phytophthora. And what we showed is that 4 mutations in the ligand binding pocket are sufficient to change its binding specificity, and it reprograms it into a very high affinity receptor and sensor for this molecule. When we put that engineered receptor back into transgenic plants, we've done this in lots of plants, this is just Arabidopsis, we're able to activate ABA responses when we treat them with this chemical. So that was our first hint that we can reprogram the ligand binding specificity of this receptor. That sort of thing has been done in lots of receptor systems, but what we observed when we were doing this was something that was, I thought at the time, a little peculiar, but that we didn't really make a big deal of in the paper. So the way we identified that receptor was we did saturation mutagenesis of all the ligand contacting residues in the ABA binding pocket. So that means for each residue, we made all possible substitutions. So, it was, I think, 19 residues by 19 mutations at each residues. And then we systematically tested each of those mutations using our yeast 2 hybrid type assays against a panel of agrochemicals that were non-herbicidal. And we were just looking to see, can we activate any of these mutants in the pool with any chemical? And this is just a summary of some of the data. Along the top is the specific amino acid residue that's being mutated, and then along this axis are the specific mutations that we're testing. And if a box is filled in, it means that single mutant responded to one of the 16 chemicals that we tested in our system. So we tested 16 chemicals, and we were able to get single mutants that responded to 4 of those. And these are structurally unrelated molecules. They don't share some common scaffold. I don't want to get too far into the weeds, but what we did in the system was we started at really, really high concentrations. So these were not good receptors, but they were still responding to some level to the compounds. And then the final receptors we got were through iterative mutagenesis and selection, a kind of standard directed evolution pipeline that got us down to high affinity sensors. So anyway, the bottom line here is that what was surprising at the time was that it worked for so many different molecules that looked nothing like ABA and didn't look anything like each other. We didn't really know what to make of that at the time. So the question really was this a fluke? Would 25% of any small molecule that you put through our system work? And can we improve it? And so really part of what started that project that I started the talk off with was really a belief that no, it wasn't a fluke, and that yes, we can improve it. And I think the final answer is that's true, but it's not going to work for any molecule that we want it to work for. Okay, so the project, and I won't think conceptually this is very similar to a lot of what I've already told you, so I'm not going to go into all the details, but I will tell you the difference that we did the second time around. So the first time around, we started with a saturating collection of mutations in the binding pocket. The next time around, we collaborated with a talented protein engineer by the name of Tim Whitehead at the University of Colorado. And instead of making single mutations, we used some methods that his lab had developed to systematically make all possible double mutations in the ligand binding pocket. We didn't make them all, actually. Tim's lab used the Rosetta protein design package to filter out any single mutants that were predicted to be destabilizing. So that allowed us to create a library of close to 40,000 mutant receptors that each had, double mutations inside their ligand binding pocket. And this is a much bigger number of mutant receptors than we had screened in our first iteration. This was built using oligo pools. So these are pools of synthetic oligonucleotides that you can design on a computer. And the method that Tim's lab to develop this, probably we wouldn't use anymore. If you're curious, I can tell you the technical details. It's now much more efficient to build these types of libraries using oligonucleotide pools and Golden Gate assembly. Nonetheless, we were provided with a collection of close to 40,000 putatively stable double mutants, and we put them through this pipeline that we had developed earlier, screening for using yeast-based assays to see if any of the members of the library would respond to a panel of target compounds. We focused on some substances of abuse and some banned pesticides. We screened about 38 molecules from these classes, and at the end of the day, there was some rounds of mutagenesis and selection to optimize the receptors that we got out. But at the end of the day, we wound up with sensors that were fairly high affinity from low nanomolar to low micromolar for a fairly wide selection of compounds. Probably the first thing you're thinking, so one of the reviewers was like, you're just mutating a critical specificity residue, and you're really just making turning the receptor into a kind of non-specific entity that will interact with many small molecules. And so we don't think that that is the whole explanation. There may be cases where that's true, but what we've seen from characterizing the selectivity of the receptors that came out of these are a couple things. One is that as we go through the rounds of iterative rounds of improvement through our directed evolution pipeline, we get to really high affinity receptors. When we profile the selectivity of those receptors over the course of the evolution experiment, it's clear that the selectivity is improving as we improve affinity. That's a fairly common feature in a lot of biochemical systems. We've also done things like engineer receptors for closely related compounds. So these are two receptors that recognize two different organophosphate insecticides. They're not identical, obviously, but they're quite similar. What's being plotted on here is how well that mutant receptor binds to and inhibits the pink blob from earlier, the phosphatase. If it's activating it, we get close to 100% inhibition of the phosphatase activity. And the blue molecule here is the on-target ligand, and the green plot here is the off-target ligand. And so the right receptor is responding to the right compound. We don't have perfect selectivity, but this is a log transformed x-axis. So these are really very selective for the molecules and can discriminate between closely related molecules. So I won't, this is published it's about a year and a half ago now, so I won't go through all the details. We understand the structural basis for some of the receptors that we've engineered through work with structural biologists. And we've shown that we can use these receptors as generic chemically induced dimerization modules for triggering different types of outputs. Like in this example, we're getting nanomolar EC50 values or activation values for a luminescence assay that's based on our system. This is a transcriptional circuit. We've built sort of simple ELISA-like dot blot test systems with it. But that is not so much what I want to talk about here. So there's one problem with that experiment that we did, and that was we were really not very biased in the way we approached it. We weren't very unbiased in the way we approached it. Put another way, we were quite biased. So we kind of focused in on classes of molecules where we had some evidence already that they would work. So we had like identified one of these cannabinoids as a sensor, and so we screened a whole bunch of them. And so this is a heat map that is illustrating the pairwise similarity of all of the compounds that we screened in that paper. And if it's blue, that means it's perfectly identical. If it's farthest to red, then there's no detectable similarity. And what you can see is that there's this big cluster of molecules that are kind of yellowish and bluish. Those are all synthetic cannabinoids that are closely related to one another. And they came up, as hits. So to cut a long story short, we wanted to really address in a more unbiased fashion if this works and how well it works, and to put some parameters on the process. So a former postdoc who now has his own lab in Delaware, Jesus Beltran, set up a high throughput version of this screen. And because I want to get to more of the plant stuff in the next few slides, I'm just going to give you the big takeaways. We screened a collection of about 2,800 FDA-approved drugs and natural products and agrochemicals and herbicides. And we developed a kind of high throughput version that would allow us to go through large numbers of compounds very quickly. We got a ton of receptors for many different compounds. There are kind of clusters. So there's, for example, quite a number of steroids that we were able to develop sensors for, as well as quite a lot of natural products. The final analysis is that about 6% of the small molecules that were present in the collection of molecules that we screened, for those 6%, we were able to identify mutants that recognize one of those compounds. So in kind of one high throughput experiment, we were able to isolate close to 200 biosensors. And that is qualitatively very different than any other sort of receptor engineering project that has been done. And so this is just a little table from a review from George Church, and it's just summarizing some of the many types of sensor and engineered binding pockets that have been developed. For the most part, the strategy is you take an existing transcription factor that binds to a molecule like lactose or a closely related molecule, you engineer it, and usually what you can get is a new receptor that maybe recognizes an analog or something a little more distant, but you're not usually getting hundreds of molecules that are unrelated to one another like what we saw. And so what we would like to do here is to create this, improve these systems so that we can make isolating these types of small molecule regulated switches almost as easy as it is to make an antibody against a protein. So this would be for small molecules, and there are some caveats around this. The molecules can't be too small and they can't be too big, but they're definitely the class of molecules in what's called the drug-like space, which is the things that includes known drugs as well as experimental molecules that could become drugs. Okay, so why does this work? Why was it different than many of the other kinds of systems that I talked about? So there's a really interesting feature of the ABA receptor as well as the gibberellin receptor, one of the other plant receptors that work in this induced dimerization system. And that is that there is what we call affinity amplification. There's really two KDs that are critical to the overall sensing that's going on. There's an initial KD that involves the binding of the ligand to the receptor to close the receptor and form that activated conformer. But then once the activated conformer forms, that phosphatase, the pink blob in the earlier cartoons, binds to the activated receptor. The ligand is fully bound inside the receptor, so it's not solvent exposed. The phosphatase isn't seeing the ligand at all, at least not directly. It's recognizing the closed activated form of the receptor. And that second KD for the binding of the phosphatase to the activated receptor is extremely low, high affinity with a low nanomolar KD. So really, we have a fairly weak initial ligand binding interaction with the receptor that's converted into an overall high affinity sensing system because of this secondary binding event that's extremely high affinity. And this is probably why the engineering has worked so well, because we're starting off screening chemicals in the 100 of micromolar range. So those initial receptors are fairly weak. They're probably, if you could measure the KD, we guess that they would be very, very high micromolar or low millimolar. But we can detect them in our yeast assays because we have this amplification system that's provided by the high affinity binding of the phosphatase to the activated receptor. Okay, just checking my time here. All right, so what I've told you up to now is that we think we can really reprogram the binding of this receptor and use that to create all kinds of drug-regulated switches that could have applications in all of biology, not just plant biology. But we haven't got to the plant part, which I was saying is a problem, because if we put these receptors back into plants without changing them any further, they're just going to activate ABA signaling because they're going to bind to their endogenous pink blobs and trigger the signaling cascade. So this brings us to the point where what we focused on was trying to rewire the interface between the two proteins to create a signaling module that functions orthogonally to the native signaling module. The work that this work just came out a few months ago in Nature Chemical Biology, so you can look to that paper if you want to get some of the details. I'll just try to stay focused on the high level stuff. So basically, with that first story I told you about, with this engineered PIR1 MANDI receptor that we put into plants, it was able to activate signaling because when that ligand bound, it would form a complex with the phosphatase and activate ABA signaling. So what we wanted to do, or what we set out to do, was to introduce mutations into the receptor that would preclude it from binding to its phosphatase partner, and then introduce compensatory mutations into the phosphatase that would preclude it from interacting with the wild type receptor, but would restore an interaction with this otherwise non-functional receptor. So basically like a bump in a hole type strategy where we were adding a bump here and making a hole there. Actually, as I'll show you, what worked was introducing a salt bridge that was essential for stabilizing this complex and destabilizing interactions with the wild type partner. Another component that was necessary was that we had to eliminate the phosphatase activity of the protein we were engineering here, because we don't want to dephosphorylate any of the downstream targets. So once we had this module, the first thing that we wanted to do with it, which I'll show you data for in a moment, was to build transcriptional circuits that we could then go and reprogram the ligand binding specificity for, so that we could engineer those kinds of plants that I started the talk off with that would tell you if chemical X was present in the environment. But there's many other applications as well. So I guess I do want to make sure there's enough time for questions. So you can look to the paper if you want to get into the weeds. The short version of this is we did, I'm a kind of geneticist by training, and so we did a lot of genetics. We created a collection of mutants of the receptor that wouldn't bind the phosphatase, and a collection of phosphatase mutants that wouldn't bind to the receptor. And then we did a genetic selection to look for mutually suppressive or restoring mutation pairs. And that led us to this salt bridge that gets introduced into the protein and allows this orthogonalized signaling module to interact, the two pieces to interact with one another, but the two mutants won't interact with the wild type partners, where obviously the wild type proteins will. The mutations that we introduced, as I said, create a salt bridge, And we think that that is really sort of the secret sauce of what made this module fully orthogonal. We showed that we could take those mutations that had given us sensitivity to that agrochemical Mandy earlier that I told you about, and we could graft those into this new system. We call it the Pure One Star system, and we could engineer a new module that would respond to that agrochemical, and the pieces that we had created, similarly as I just showed you before, wouldn't cross-react with their wild-type partners. So this is just to say that it is a really good orthogonal system where we can change the ligand binding specificity. And to bring this stuff together, if we want to get to that vision that I started off with, we're going to need to be able to reprogram the ligand binding of the new engineered receptor as well. And so we did a pilot screen of that. We haven't gone as far as we have done with the other scaffold, the wild type starting point that I told you about, but we screened a panel of the same types of pesticides that we had screened with the wild type receptor. And what we found was that we were able to get receptors for all of the same ones that we could get the normal receptor libraries that we had made. And in particular, we focused in on making a high affinity receptor for one of these molecules, a banned organophosphate pesticide called azinphos ethyl. And so Jingda, the postdoc that did this work, this is just the biochemical or the yeast-based data showing the mutations that are present in the receptor, the low nanomolar responsiveness we're able to get in the yeast systems, and the orthogonal behavior of this newly engineered receptor. So what Jingda did was he created a simple transcriptional circuit where the orthogonalized phosphatase is expressed along with the engineered orthogonalized receptor. They're fused to the Gal4 DNA binding domain and a transcriptional activation domain. So when those two pieces are expressed and come together, they can bind to this UAS that's driving expression of a gene called Ruby. Ruby is a synthetic plant gene that's exploded in the plant world recently because it allows, with a fairly concise biosynthetic gene, to produce the beet pigment, one of the beet pigments, the betalanes that make pigments beet red. So when he put this circuit into Arabidopsis, what we saw, much to our satisfaction, was that we were indeed able to engineer plants that would sense this starting molecule that the plants never sense, and to do it in a way that's really easy to observe through simple visual inspection, which was one of the original goals of that project that got us going down this path. And this is just to show you it works in independent lines, and actually we can very reliably start to see indication of the chemical's presence in the low nanomolar range. Okay, so I won't talk a lot about, I'll tell two little short stories about what we've done with it. One, and not say too much more, but we're gearing up to do a lot more. One is that a collaborator at the Danforth Institute, Meter Nusenau, has taken the pieces that we built and used them to see if this will work in monocots. And his hands were able to use this to drive expression of reporter genes in Ceteria. The other thing that we've done is I have a whole separate project in my lab that I don't have time to talk about that's new, that I'm excited about, that's like a speed breeding project for citrus. And so we've used this system to build a genetic circuit that turns on FT. And the key here, the trick or the secret sauce to this is that we're turning on the expression of the FT gene with one of these approved agrochemicals. So it's something that's really well suited to being used in a crop or in the real world outside of the Petri plate environment. And so we're able to get diverse citrus germplasm flowering and go seed to seed in just around a year with really pretty minimal phenotypes if there's no treatment or induction. And so that's another project that's going on, but it built on these tools. And I think there's just a lot of things, similar things that we might start doing. Okay, so this work is continuing, and we have a whole new project that I'll advertise in just a moment. But I will say that one of our other dreams is to create more than just one of these orthogonal signaling modules. So we would like to be able to control 2 or 3 different channels in a plant, and to be able to control the ligand specificities of those 3 channels. And so we've started on the track of making another one of these modules that seems to be working, but we haven't characterized it quite as extensively. Okay, so just to put this all together, my lab started off with this interest in understanding how ABA works. And we discovered that, ABA's got this really interesting chemically induced dimerization mechanism that's driving all of its perception. That the features of this biochemically, we think, enable us to rewire the ligand binding specificities quite efficiently and effectively. And that we can reprogram this module now so that it's able to act as a generic circuit for plants. It could also be used in any other system. We've put it into E. coli and controls proteins in coli. These should be fairly generic building blocks that can be used across kingdoms. Okay, so these are the main contributors. I think I've acknowledged almost all of them as I went. I forgot to acknowledge Sung-Yul Park, who really is the main protein engineer in my lab and has been a member of my group since I started at Riverside. I acknowledged all the other contributors who showed us data. And before I take questions, I'll just say that this project, they liked it. And we applied for they don't renew grants, but they create new programs. And so the new program that they're interested in is in trying to engineer similar types of indicator plants, but where microbes in the soil would be responsible for sensing things, and that those microbes would then send signals to the plants that trigger secondary signals that allow for the events sensed in the soil to be perceived in the outside world by a naive observer. And so we've got a big project that involves lots of labs. Many of the pieces rely on the technology we've built here for engineering chemically regulated switches that work in both microbes and plants. There's a lot more pieces to this project. I'm happy to tell you more if you want, but we need people for this project. So if you're looking for a lab out there, send me an email. It's a super exciting project. And with that, thank you.
¿Preguntas?
Hi. I have many questions, but I will make 2. One is related to the ABA receptors. So just to make my mind clear, there is only this type of receptors for ABA, the pill-like receptors, or there are other receptors, because I remember that there were like membrane receptors for ABA, and I don't know if still that is maintained like a true fact or not.
Do you want me to answer that question, or you want to do your second one?
No, that question, because the other one has to do with it.
All right, so yeah, so the part of the story I didn't tell was that when we published our paper in 2009, back to back with another lab that independently found the same receptors and came to many of the same conclusions, there had been a few papers claiming that the ABA receptor they'd identified the ABA receptor. One of them was retracted. One of them probably should have been retracted. Another one, I don't know. Maybe it's these GPCR-like proteins that have been proposed to bind ABA. I don't know what to say. Like, we haven't worked on it. The phenotypes, when you knock those out, are very weak. And what we know genetically, and my bias is from a genetic perspective, is that If we knock out the 3 major SNRK2 kinases that our receptor family works through, those plants are barely viable and they're completely blind to ABA. So I would say if there's some other pathway that's not interacting directly with the core, what we call the core pathway, its effects are very minor or restricted to some part of development that we're not familiar of. There very well could be other ABI binding proteins. Some people have pulled Rubisco out, I think got pulled out in one ABA binding experiment. So if you define receptor as a binding protein, there may be many receptors. I think the more interesting biological question is, are there other really, really important central nodes that regulate ABA signaling? Yeah. There's 14 ABA receptors. Jinkong Zhu's lab has knocked them all out. Those plants too are barely viable and blind to ABA. So I think that the majority of ABA signaling goes through that pathway. That's not like I'm not the only one who says that. I think that's the consensus.
Okay, thanks. And the other thing is, there are more than one phosphatase in the plant cells, right? So when you modify one, still there are other phosphatases that could recognize the modified receptor. Do you have that interference or not? There is not mine at all.
We haven't done pairwise. We haven't tested every phosphatase with our engineered receptor. But the mutation, that salt bridge that got introduced, is in a pretty conserved residue. And we've overexpressed both pieces as GFP fusion proteins in Arabidopsis. And we don't see, 8 different differences in ABA sensitivity. That's kind of in the supplemental data to that paper. So there's probably other things they could bind to too, but we don't think that we think they're sufficiently insulated from the native ABA signaling pathway that's not something we have to worry about. But yeah, that's a good question.
And the last one was the last one. The last one has to do with the sensitivity of this pair. It's supposed to be the KD is a very high KD, so it's very high affinity, very low. It's the other way.
Yeah. I put that. Sorry, I confused that earlier.
Yeah, there is a high affinity for this. No? So when you are planning to do these detectors for different agrochemicals, if the affinity is very high, then it would detect almost very small amounts, which are not maybe relevant for whatever they are using in agriculture.
Yeah, so the question, just to summarize it, is that we might not always want high affinity sensors, right, is what you're saying. So there may be cases where you want, like if it's a nutrient, for example, you may want to be sensing in the millimolar range or the micromolar range. So we haven't done this yet, but we can see, like, it's tunable. There's different ways we can tune it just by the number of mutations that are present, but also the affinity of the receptor for the phosphatase can be altered by mutations at the interface. So I think that should be a solvable problem, but it's a very good point that we need to factor that in if we're going to really try and make sensors that are useful over a broad range.
Solo recordarles a las personas en Zoom y YouTube que pueden escribir sus preguntas y aquí las leemos.
Thank you, Sean, for the talk. It's very nice. I have a question, 2 actually also. The first one is, when you apply this agrochemical, how stable is this reprogramming, and how long it will take to go to normal?
Okay, so that will be very ligand dependent. So the Mandy, that agrochemical that we repurposed, that is actually one of many products that are used for controlling Phytophthora. And its kind of selling point for farmers is that you only really need to apply it once or twice. It adheres to the cuticle and it's very long lasting. So that's probably not going to be super reversible if that's what you're getting at least on short timescales. But I think that what excites me about our system is that I think that there are cases where there are specific molecules with specific properties that you may want for an experiment. So you may want something with a really short half-life, and we could conceivably design a circuit that's regulated by a molecule with a really short half-life that kind of logic. But for that one example with Mandy, that's probably pretty stable and not good for a short-term kind of induction and going back.
And that's comes from the second question, because actually ABA, of course, you show with the phenotype that they are involved in the open and closure of the stomata, but it's also involved in many other processes. And then if it's really stable, this interaction or this reprogramming, they could have a side effects or pleiotropic effects that not are very well desired.
So yeah, for like the ABA, for the Pier 1 Mandy piece that I got started talking on, we actually apply pretty low concentrations, so like micromolar, low micromolar, much lower than we would 1 micromolar, which is substantially lower than we would use with ABA. And they definitely go back. They go back to normal, but there's a persistent ABA effect, much more than if you just sprayed with ABA, for sure. Probably because of its half-life being longer, I guess. Yeah.
Thank you.
By YouTube, somebody asked, is the accumulation of ABA in plants primarily a short-term drought tolerance strategy, or does it also play a role in long-term responses that can induce to lethargy or dormancy?
What was the last?
It's a long-term or short-term response to ABA.
Right. Did they say dormancy?
Yeah,
okay.
Dormancy.
So for the most part, I would say ABA is a short-term molecule. It has a really short half-life. It's been measured. It's on the order of hours, not days. But there are some developmental effects that would persist after the hormone levels go back down to normal. So there's a recent paper showing that this pathway is kind of interacting with the pathway that influences stomatal development. And ABA mutants have differences in their stomata and stuff like that. So there can be longer term effects that are caused, or root system architecture is affected by ABA. And so that once that's set, it's going to last for a long time. But I don't think it's the plant's long-term answer to drought. Those are plants have come up with different solutions. I think it's for short-term mediation of changing environmental conditions, both locally at the leaf level and then the soil, roots in the soil. That would be my bias, but there are papers that have tried to suggest kind of longer-term epigenetic effects that might persist, and they may be real. I don't know, but my take is it's short-term.
Yeah, here. I really like it, your talk and the approaches. But then some of us have a problem that we don't work with Arabidopsis, that is easy or somehow easy to have a stable transformance and to do this kind of approaches. Do you know if there is any other approaches where you can somehow introduce these proteins, these modules into other plant species?
Well, do you mean without making a transgenic?
Exactly, yes.
I think you might be hitting a roadblock there. I'm not aware of any way to do that. Sorry. I know it's a pain when you can't transform your organism.
Over here. Oh, thank you for your talk. I was just wondering about the location of the receptors. I mean, as far as I understand, this molecule should get inside the cells, but about the location, well, I was just thinking that maybe if there's any receptor that is located outside the cell or on the membranes, and a way to engineering the mechanism of catching the molecules, not just inside. Or maybe.
Do you mean for the ABA receptors or just in general?
In general, I mean, for what where are those the receptors?
So in general, I think that for small molecules, the kind of things we're going after, drug-like molecules, they're usually getting into cells through passive mechanisms. So I think maybe for like a peptide or something that's too big to diffuse, to get across the membrane, you might want in those cases to have a membrane anchored receptor. I think the engineering challenges there are substantially greater, And in plants, it's not, maybe there's a few like scaffolds you might consider using. But I think that would just be a whole different idea, you know, that we're focused on kind of the drug-like space for at least for the controlling ligands. But yeah, it's a good idea.
It sounds like a really great challenge to do that, but thank you.
You're welcome.
Hey, that was a great talk. I was wondering if your lab is interested or is making efforts to detect endogenous chemicals of the plant so that you can detect, like have sensors for endogenous molecules, and whether the readout could be reprogrammed for, I don't know, modifying the root system architecture or some developmental readout control that might indicate or make a record of where these chemicals were at a certain time and period.
Yeah. So we definitely are thinking about that. I didn't mention this work. I'm sure you're familiar with it, Cesar. That you can fuse the two pieces to, you know, a fret system and use the ligand-induced changes in fret to detect ABA levels in plants. So these have been used, kind of rewired to make sensors of endogenous ABA levels and look at their levels at different parts of the plants. That's from Wolf Frommer and Alexander Jones. So I think it should be possible. Ruby turns out to be actually a kind of interesting marker. We've found that it doesn't, at least in Arabidopsis, it doesn't ever go away. So we have plants where we have almost like zebra stripes from applying the chemical, getting ruby made, then they start growing and there's no more ruby. And so I think actually that the betalanes are possibly interesting for time recording, but developmental things could be possible too. Yeah, we're thinking about it, but it's not necessarily the next thing we're doing. A lot of the molecules that came out of the big screen I described are plant natural products. They're not necessarily, they're somewhat usually specialized to specific species, but we could use this system to look at where they are and how they're help, you know, identify transporters and things like that.
Okay, thank you very much. My question is about this new project. So, well, there are actually two questions. The first one is, how do you select the bacterial population that is going to be sensing the soil? Because, well, I think that there are two principal factors. It has to be symbiotic with the plant, and also it has to proliferate even in the presence of the soil contaminants that you're sensing for. And my second question is, how can you make sure that even if if something happens with the soil, the composition of your population in microbes will stay the same?
You're putting your finger on one of the really major challenges of the project, and I think that there are many goals. I framed it in kind of a sensing context, but I think that the other real big challenge and hopeful goal of this project is to come up with rules for building stable synthetic communities of microbes that aren't competed out by native microbes. And that, you're absolutely right, it's a hard problem. Part of what we'll be doing is isolating microbes and trying to build synthetic communities. We have one of the collaborators on the project, Becky Bart, has done work like that in other organisms. So we're doing a kind of trying to reconstruct and build synthetic communities. We may work with endophytes, you know, things that are going to be taken up or can get packed, you know, cracks in the roots and start growing inside. But we have, you know, approaches that we're going to take, but we don't have a real set of microbes yet to do that. There's lots of pieces, and yeah, part of the project involves sort of incrementally increasing, you know, how long the microbes live, because they're probably going to be outcompeted fairly quickly in the beginning. Yeah, so I don't know if that answers your question, but it is a good question and a big challenge.
Hi, Sean. Thank you very much for your talk. My question is regarding some technical issues you can encounter in using this system as a receptor. So I was wondering, how long does it take to the plant respond to signals, or the systems in general to respond to signals?
Well, like in that last slides I was showing with the ruby, that was within 48 hours. That's, not, I'm sure that there was responses earlier. In citrus, we've done like, qPCR, and within hours we can see induction, but like induction of flowering takes a couple weeks. It requires actively growing shoots, before you actually see a meristem that's floral. So it's going to depend, but at the transcriptional level, it seems to be fast. It will depend on a lot of factors, like the chemical, the way you apply it, if you do it in liquid culture or whatever, but it can be very fast.
Okay, thank you very much.
You're welcome.
¿Alguna otra pregunta? Okay, si no, Sean, thank you very much for your very nice talk.
Thank you. I had a wonderful visit.